Welcome to your first Patho lecture. So in this video, I'll be doing Chapter 1 and 4. Chapter 1 you'll find is pretty basic information, where Chapter 4 will get into a little bit more of the biology, but again at a very basic level. So there is an expectation here that if you work through that lecture and you aren't understanding all the content, that you go back into the text and review. So let's start with definitions. World Health Organization, or WHO. This is one we've all heard of in the news, especially with Ebola and SARS and things like that. So the World Health Organization defines health as being a state of complete physical, mental, and social well-being, not merely the absence of disease and infirmity. So it's not just that you're not sick, it's that you're actually well. The World Health Assembly, its definition is that all citizens of the world reach a level of health by the year 2000 that allows them to live a socially and economically productive life. Public Health Agency of Canada, health is determined by complex interactions between social and economic factors, the physical environment, and an individual's biology and behavior, plus governmental policies with the universal access to health care. Long story short, health is a very complex definition, because it encompasses so many different aspects of our lives. When we look at pathophysiology, it's really a combination of two fields, pathology and physiology. Now I know most of you will have done physiology prior to this course. However, if you're in the ASP program, it is not required of you. So if you don't have physiology, you're going to have to depend a little bit more on the text, where if you have physiology, you'll be a little bit ahead. So physiology, by definition, is the study of body function, where anatomy is the study of the structure of the body. Pathophysiology, again, is the combination of physiology, how things work, and patho, or disease. So this is the study of the body's response to dysfunction or disease. So disease is defined as an interruption, cessation, which means stopping, or disorder of a body system or organ structure. Terms that you absolutely must know are etiology, pathogenesis, morphologic changes, clinical manifestations, diagnosis, and clinical course, and we'll be defining them shortly. So we need to understand this basic terminology within pathophysiology, just as we did in anatomy and physiology. One thing we have to consider when we look at pathophys is the idea that everyone is going to adapt to the changes in their body differently. And these stressors can be physical stresses of a disease, they can be emotional, they can be environmental, but there's various factors that are going to affect that reaction and how well you can adapt to it. Age, health status, psychosocial resources, the rapidity in which the need to adapt occurs, so basically how quickly things are changing in their body. Do we have 6 months to adapt to that change, or do we have 6 hours? Also, the availability of adaptive responses and the ability of the body to select the most appropriate response. In other words, do we have a built-in response in our systems that will help us compensate for the change? And is our body able to choose it? So here are some definitions. So etiology, simply the cause of disease. That's one you're going to see a lot. Pathogenesis. So here again, if you've had a course with me before, you'll know that I love to break down words. So we have patho and genesis. Patho is disease, and genesis is formation. So if you know those two factors, you know the definition here, which is the process of the formation of the disease, from the initial contact until the expression of the disease. So morphologic changes are talking about morphology, or what we look like. So these are gross anatomic or microscopic. So we can either see them with our naked eye or under the microscope. So clinical manifestations, signs and symptoms. You should be able to differentiate those two. Where signs we can see as an external observer, things like a rash, redness surrounding a laceration, where symptoms are coming from the patient. So not something that we can observe as an outsider, but rather the patient explaining that their ankle is sore, it feels hot, they're uncomfortable, they have a headache, they're seeing double, things like that, something that we cannot physically see. The diagnosis is the designation as to the nature or the cause of the health problem. So what's actually causing it? And lastly, clinical course, which is the evolution of the disease. So if we go back to the first one, we're going to talk about the causes of disease. When we think about etiology, or the cause of disease, the causes will fall into 4 categories. So biologic agents, bacteria and viruses, physical forces, trauma, burns, radiation, chemical agents like poisons or alcohol, and nutritional excesses or deficits. But we need to also talk about the methods in epidemiology, which is the study of the spread of disease. There we go. Whoops, too far. There we go. So epidemiology is going to look at how disease is spread, how to control it, how to prevent it, how to eliminate it. And if you followed the Ebola breakout, you really have an idea of what epidemiology is. Epidemiologists, excuse me, had a huge role to play. We had to have a very good idea of how that disease is spread. Is it physical contact? Is it airborne? Is it going to spread easily over large distances, or do you have to have physical contact with the infected person? And if so, do you need to have contact with a bodily fluid, which is what the case was with Ebola. Knowing that will help determine how to control the disease and also how to prevent the disease. So how to control the outbreak that is already there, how to prevent further outbreaks, and overall, how to eliminate it altogether. In order to really understand the disease, we have to understand what's causing it. So in the case of Ebola, it was a virus. So again, the diagnosis is a designation as to the nature or cause. When we're looking at these, we're talking about predictive factors. So really kind of attaching characteristics to our knowledge. So the reliability. So the extent to which an observation, when repeated, gives the same results. So if we see a certain rash, does that mean that this individual has a certain infection? And how reliable is that? Does it happen 90% of the time? Does it happen 5% of the time? What about validity? So when you see validity, think about valid. So we're talking about how valid a measurement tool is. So again, if we look at a rash, right? If we have a measurement tool, say it's a computer that analyzes an image or a blood test, how valid is it? How accurate is it? And that leads us to sensitivity. So sensitivity, we're talking about true positives. So how many times does the test give us a positive as an outcome, as a result, when the individual is actually positive? And so the easiest way to understand true positive and true negative, I always go back to pregnancy tests because it's super straightforward. So for example, when you're talking sensitivity, what percentage of the time would the test give you a positive when the individual was actually pregnant? Specificity is telling you, again, true negative, how many times would that test give us a negative when the individual was actually negative? So there are two different things there. Sensitivity is how sensitive is it to detecting that condition? In this analogy, it's pregnancy. Specificity is talking about how specific is it, right? How often do we get a false positive so that the test sees a positive when the person really isn't pregnant? So again, it's about being specific and not getting that false positive. So those are two ideas, sensitivity and specificity. Again, you should be able to define, but also differentiate from each other. Lastly, a predictive value. So the extent to which an observation or test result is able to predict the presence of a given disease or condition. So again, going back to blood tests or urine tests, how well is it able to predict what's going to happen or the presence of the disease? All of these tests are based around a normal. And so we have to talk about what the normal is versus abnormal. So the normal values are established based on a sample population. And so what you're looking at here, when you look at blood values, the example here is sodium, what you're seeing is a mean, and usually there's a range here. And what you're seeing is mean plus or minus 2 standard deviations, which is giving us a 95% confidence interval. If you've done statistics before, you'll understand that. And so if you haven't done statistics, here's the deal. I teach stats too, so it's not my favorite subject, but it's good for helping understand all of this. Essentially what we say is that the range given here, excuse me, so for normal sodium, 135 to 145 millimole per liter. When we look at that interval of 10 millimole per liter difference between the lower value and the higher value, what we're essentially saying is that this represents 95% of our population, of our normal values from our population. So not individuals who are known to be hypertensive and have high sodium, not individuals that have lower sodium. This is the normal range for 95% of the population. And that's what that confidence interval is. We're confident that if we repeated it 95% of the time, it'll fall within that range. That's really what it comes down to. One thing that we have to be mindful of is that our normal values may vary by age, sex, or even health status. So again, if someone is hypertensive, they're probably going to have a higher than normal sodium value, right? If they are dehydrated, it's going to change as well. And so we have to be conscious of how sex and age or health status may change this normal. When we look at testing, we're going to talk about false negatives, false positives, true negatives, and true positives. And this is really what we were just talking about a moment ago. And so, again, the idea that if the test says it's positive, so if the test says it's positive and the disease is actually present, it's a true positive. Where if the test says it's positive and the disease is actually absent, it's false positive. So think of this as a cancer test or a test for pregnancy, a test for diabetes, anything really. It doesn't have to be a disease necessarily. So if the test says it's negative, but the disease is actually present, it's a false negative. Where if the test is negative and the disease is absent, it's a true negative. So if you have a positive result on the test, but the disease is actually absent, it's called a type 1 error. If you have a negative result in the test and the disease was actually present, it's called a type 2 error. Now this is something that I'm not going to test you on. There might be one multiple choice question on. It's not something that is key to this course, but it is important for you to understand what a true positive, what a false positive, what a false negative, and what a true negative are. Because if you move on into any other human biology course, especially in a medical field, these are terms you absolutely must know. And these are related to the positive predictive value and the negative predictive value. So again, the positive predictive value is the portion of true positive results, and that's related to the sensitivity we saw before. The negative predictive value is the portion of the true negative results, and that's related to the specificity that we talked about before. So we're moving on to the next part, and that's your clinical course. When we talk about this, we need to talk about all of the different designations we give disorders. So an acute disorder is relatively severe, but is self-limiting. So in other words, the disease limits itself. And we'll see examples of that as we move forward. A chronic disorder, chronic is always talking long-term. So this is long-term, it's continuous, and it can include exacerbations or remissions. So it can just be continually getting worse, or you can have these fluctuations, this kind of roller coaster ride of exacerbations when you get worse and remissions when you get better. And you can fluctuate up and down. You can also have subacute disorders. So sub means under or below. So a subacute is in between the two. So it's less than acute, but it's not quite chronic. So this is an intermediate or between acute and chronic. It's not as severe as chronic, hence the sub, but it's not as prolonged, it's not as severe as acute, but it's not as prolonged as chronic. And again, sign, symptom, and syndrome. Sign is something that we can see as an external observer. Symptom is something that only the patient can tell us. And a syndrome is the compilation of the two. When we think about all our different conditions, we need to talk about risk factors. And in order to determine what our risk factors are, we have a number of different kinds of studies that we can do. One is a cross-sectional study that uses simultaneous collection of information from a number of different groups in order to classify exposure and outcome. Then we have case control studies. You've probably heard about case studies. And so here what you see is you compare case subjects to control subjects. So you see individuals who were exposed to a certain pathogen and those who weren't. Lastly, a cohort study. So it's going to involve groups born at the time or who share some interest. So the Framingham study is the largest that we've ever seen. It's 5,000 people, aged 30 to 59, and they're followed for 20 years. It's an enormous study, and we've learned so much from it. And if you look in your text, you'll see all of the amazing findings that we've learned from that one study. And if you follow that link, it'll take you right to all the data, so you can play around and see what you can see. It's really amazing. So when we talk about disease frequency, we have to talk about individual cases, whether they're new or whether it's just a new episode. We need to talk about incidence, which is the number of new cases, and prevalence, which is the number of existing cases in a population. So incidence can be something referring to, SARS is something new coming in, where prevalence can be something like measles, which is always around to a certain level, but the number of cases will fluctuate based on vaccination rates, time of year, things like that. When we talk about these terms, we're getting into the details here, especially for the epidemiologists, which again is describing what the disease does. So morbidity is talking about how the illness really affects quality of life. So it's going to be concerned with the incidence, the persistence, and the long-term consequences. Mortality is going to give us more information about trends in health of a population. And mortality is talking more about lifespan. The prognosis is the probable outcome and the prospect of recovery. So you're talking about whether or not this illness will be terminal, whether the person will recover fully, or whether there will be long-term consequences. When we look at the natural history of a disease, we need to be talking about what happens if the disease runs its course, and just kind of the basic process. What's going to happen first, second, third, fourth, fifth. So this is used as a predictor of outcome for diseases for which there is no effective treatment. So again, it's natural history of a disease because we're not trying to alter it. So the information about this has a lot of potential for effective treatments, because once we have a clear understanding of what happens, we then have a better idea of what aspects that we can target for treatment. So for example here, Hep C, so hepatitis C, 80% of those who become chronically infected cannot clear the virus and progress to chronic infection. So in that case, we have to talk about the natural history of the disease, what's going to happen next, in order to have a better understanding of what that patient is going to progress through in different stages and how we can best intervene. The best case scenario, however, is preventing diseases altogether. So primary prevention, immunizations. And what you'll find as we work through this course is that I'm a strong believer in immunizations. And I'll show you a link in a moment that started all this anti-immunization, anti-vaccination movement. So primary prevention, immunizations. Essentially remove risk factors so the disease does not occur at all. Secondary prevention, detecting the disease when it's still curable. A great example here with pap smears. So females, when they reach a certain age, should have pap smears done. So this is a sampling of the cells in the cervix, and essentially what you're looking for are cells that are becoming abnormal. These abnormal cells are precancerous. So if you detect those changes before they become cancerous, you prevent the cancer altogether with treatment. Tertiary prevention is preventing further deterioration or reducing complications. So here the example is antibiotic use. So again, with primary, nothing happens at all. With secondary, it happens, but it's in such early stages, it's completely curable. Tertiary, it's happened, but you can intervene and limit the negative effects on the body. So here's a great diagram kind of showing you where everything fits. So no disease, that's where primary fits. There's onset, but you're asymptomatic. You're not showing symptoms yet. And that's why a pap smear is so important, because the woman will have no clue that anything is changing, but under a microscope you can see those cellular changes. And we'll talk about what that looks like in Chapter 5 and Chapter 8. Lastly, you get clinical diagnosis. So for example, you have an ear infection, a bacterial ear infection, and therefore your tertiary is to reduce complications by the administration of antibiotics. And because I'm a mom and I know there's a million ear infections that get treated with antibiotics that don't need them, most ear infections are viral and not bacterial, and therefore antibiotics don't work. Just had to throw that in there. All right, best thing we can do is evidence-based practice. Now, if you're in nursing, you've heard this phrase a million times. If you're not in nursing, it might be a little bit new. But it is exactly what it says. We have to move forward with patients with the idea that we're constantly integrating new findings, new results, new evidence, and altering how we work with our patients. So again, it's the conscientious, explicit, and judicious use of current best evidence in making decisions about the care of individual patients. So not using information from 50 years ago because that's the way it's always been done, but rather taking the new findings, the new reliable findings, and integrating them into practice. So clinical practice guidelines will include algorithms, written directives, and a combination of both. So we may have new calculations we must do. We might have new step-by-step processes we have to follow. We might have written directives, clear guidelines as to exactly what we should do. It might be a combination of both. So again, these evidence-based practice idea will go throughout healthcare, but it's a very, very straightforward concept. So with that, we're done with chapter 1, and we're moving into chapter 4, which is really down to the biology. So with chapter 4, we're really doing a review. This should be a review of first-year biology, and if you didn't take first-year bio, high school biology even, for most of it, but certainly the anatomy that you took before taking this course. So when we look at the functional components of a cell, we're seeing the nucleus, the cytoplasm, and the cell membrane. Notice here, I'm not talking about everything in detail. I'm doing that on purpose, right? This is not a basic biology course. This isn't a more advanced biology course. And so I'll walk you through this as a review. If you need more information, more clarification, head to the textbook. It's a really great explanation. And if you're confused, as always, you can contact me. So let's start with the nucleus. The nucleus is where our DNA is. This is where all the thinking happens, where all our information is stored. This is what controls our entire cell and everything it does. So we have an envelope with pores. We have our DNA, which is compressed into chromatin. We have our RNA synthesis, and this will include messenger, ribosomal, and transfer RNA. And then we have our nucleolus. We have our protoplasm, and the composition here is water, proteins, and electrolytes. There's regions here. So the regions for our protoplasm are our cytoplasm and our nucleoplasm. And this is really just the ooey gooey stuff inside a cell. So it's what everything is floating in. Sometimes I use the analogy of a Jell-O mold. So it's kind of a 70s thing. I'm not that old, I promise. I was born in the 70s. But not that there's anything wrong with that. But in a Jell-O mold, what you would do is you would make a bowl of Jell-O, and inside that you'd stick fruit. And so a cell is kind of like a Jell-O mold, right? On the outside it gets a little tougher, so that's your membrane. On the inside you have all this fruit wandering around like the organelles in a cell, and then you have all this Jell-O-like stuff holding everything together. And so that's really what cytoplasm is. Cyto for cell, plasm for this fluid-like substance. Nucleoplasm is that fluid-like substance, but inside the nucleus. Now the cell membrane is something we're going to talk about in great detail. We're going to introduce it now, but it's certainly going to come up in a number of our organ systems. So with our cell membrane, we're looking at it being made of basically two things, lipids and proteins. So with the lipids here, we're looking at two different kinds, phospholipids and cholesterol. So our phospholipids make up the majority of our membrane, where our cholesterol is there to almost toughen the membrane up. We'll talk about cholesterol integration in the membrane quite a bit when we hit the cardiovascular system. The other really important part of the membrane is proteins. We have integral and surface proteins. So the surface proteins are only on one side, where the integral proteins transect the whole membrane. So they cross the membrane. So when you look at a cell membrane, the basic structure of it is going to be these phospholipids. But then you're going to see the integration of cholesterol, but also the proteins. So we have cell membranes, but we also have membranous barriers within the organelles. So the entire cell is encased in a membrane, but the organelles have these little barriers as well. So the cytoplasm, again that Jell-O, is a workspace for the cell. This is where you're going to get transfer of chemicals for interaction. This is where all the action happens. So here we have water, electrolytes, proteins, fat, and glycogen, and we have pigments as well. For organelles, we have ribosomes, endoplasmic reticulum, rough and smooth. We have Golgi complex, mitochondria, lysosomes, and proteasomes. So most of these, again, if you've done high school biology or first year, you'll know all these. And so again, I'm not going to go through them all individually, but just know that I have an expectation that you know the basics of these before we move forward. When we look at the skeleton of the cell, we need to talk about microtubules. And so the microtubules do exactly what they're classed as. It's a skeleton for the cell. So again, cyto for cell, skeleton for structure. So the tubules are there to develop and maintain cell form, so the actual shape. They're also going to help with intracellular transport mechanisms. So again, intra, I-N-T-R-A, is within a cell, where inter, I-N-T-E-R, would be between. These will also form basic structure for complex cytoplasmic organelles. It'll contain cilia and flagella, and these are little projections from cells that help move things around. And they contain centrioles and basal bodies. So again, really just an idea of creating the skeleton of the cell. The microfilaments will give us muscle contraction. So again, if you've done phys, you know this quite well. If you haven't done physiology, we'll look at that as we move along. So it also helps support and maintain the asymmetric shape of cells. They're thin, thread-like cytoplasmic structures characterized as thin, intermediate, and thick myosin. And so, again, all of this, I'm not going to be asking you any amount of detail on this on a midterm or final. It's really just background information that we'll be adding to as we move into other chapters like chapter 5 and 8 and beyond. So this 1 and 4 is really just to get us started. Our cell membrane is really essential to the function of our cell because it has receptors for hormones and other biologically active substances. This is how cells interact with each other. If one cell sends a chemical to the other and there's no receptors on the membrane or the membrane is non-functional, nothing's going to happen. So the membrane also is going to participate in electrical events that occur in nervous and muscle cells. So again, the idea that resting membrane potential in the nervous system that gives us our action potential to send a signal between cells, and that action potential within muscle cells, which creates muscle contraction, all of that is dependent on cell membrane. Lastly, it aids in the regulation of cell growth and proliferation. But it also does other things, like simply holding the cell together and giving us a barrier that we only allow certain things through. So with our cell membrane, we know that we have lipids, we know that we have proteins, but here we're going to get into a little bit more detail. So the lipid bilayer, so these are made from the phospholipids. This is the basic fluid structure of the membrane, and it serves as a semi-permeable membrane. So that's what I was alluding to a moment ago. Semi-permeable. It will allow some things through, but it will prevent others. So these are phospholipids with their hydrophilic head and a hydrophobic tail. So hydro is water. So hydrophilic means it likes water. Hydrophobic, it doesn't like water. So think of arachnophobic. So arachnophobic, you don't like spiders. So it's the phobic part here that you're wanting to think of. So again, hydrophobic doesn't like water, hydrophilic it does. So what happens is you have these hydrophilic heads that do like water, that face outside and inside. So outside of the cell and inside of the cell, and the tails point together. So you also have your integral proteins that'll span the whole bilayer. And so an example of these are ion channels, but there's a number of different kinds. But again, just the idea that this will allow substances to move from the outside of the cell to inside the cell, or vice versa. You also have peripheral proteins that are bound to one or the other side. So these are just bound to one side. They don't span the membrane. Then you have your glycocalyx. and this will participate in cell-to-cell recognition and adhesion. And we'll get into a little bit more detail with this when we hit the immune system. We need to talk about cell receptors and cell ligands. So when we talk about the cells and how they interact with each other and respond to changes in our environment, it's all based on receptors and ligands. So our receptors, there's two classes of them. There's receptors that use first messengers, and there's receptors that use second messengers. So with our receptors, our first messengers are things like neurotransmitters, protein hormones, growth factors, steroids, and other chemical messengers. And so these are things that have an initial reaction. They don't have to go through 3 or 4 steps in order to have a function. It's a one-step process or a 2-step process. It's very simple. The second messenger systems are much more complex. So in a second messenger system, there are intracellular mechanisms. In the first messengers, everything's kind of happening at the membrane. So that's why these are called first messengers and second messengers. So with cell communication, you're looking at autocrine signaling occurring when a cell releases a chemical into the extracellular fluid that affects its own activity. And one thing you want to notice here is that prefix auto, as auto is self. Para means around. And so these paracrine signaling cells act on nearby cells, and hence the para. They're going just to the next door neighbors. Endocrine, endo is for inside, and so endocrine signaling relies on hormones. And hormones, by definition, are chemical messengers sent in the bloodstream. They have to be in the blood. Lastly, we have synaptic signaling, which is through the nervous system. And this is what they all look like. Again, autocrine going to yourself, paracrine to a nearby cell, endocrine into the blood system. So when we look at our different receptors, we have ion channel linked receptors. So these are receptors that are going to have a very quick response, and it's because of this ion channel. So that's rapid synaptic signaling between electrically excitable cells. So again, if we want to change the charge, the electrical charge of the cell that we're interacting with, one of the fastest ways to do that is to fill that cell full of either negatively or positively charged ions. And so this results in the transmission of impulses in nervous and muscle tissue. You also have G protein linked receptors, and this is just an on-off switch for signal transduction. So we have enzyme linked receptors that are linked to certain protein hormones, and they can activate an intracellular domain. So you're just having an action inside the cell rather than just transferring something from outside the cell in. And our intracellular receptors allow ligands to move directly across the membrane and bind to the intracellular receptor. So with this intracellular receptor, it's not that the substance moving across the membrane is the action that we want. It's moving across the membrane to then bind to the receptor inside. When we look at cell division and cell cycle, we need to talk about a number of different stages, but we also have to talk about the difference between mitosis and meiosis. So with the cell cycle, the main stages of the cell cycle, we think about mitosis, which is actually the cell division part, and the interphase is the non-dividing phase. And so again, inter is between. We also know that we can have mitotic or meiotic. So mitotic cell division occurs in somatic cells, and again, where you've all done anatomy, you know somatic means general body. And meiosis only occurs in gamete-producing organs, so testes and ovaries. So the meiosis is going to give us egg and sperm, where mitosis will give us the rest of our bodily cells. So here's what you're looking at. So after mitosis occurs, and you have your two cells, you get an indefinite amount of time in G0. So this is G0 phase. So cells can stay here forever. They can stay here for 2 seconds, and they can just kind of skip over it if they want. In G1, it can be 8 or more hours, and this is where the cell is just increasing in size. It's really just becoming a more mature cell. And it's starting to get ready to go into the synthesis phase. So in the synthesis phase, we're getting the synthesis of DNA, so we're doubling our DNA. In G2, we're increasing in size, so we're getting ready to split. And then in mitosis, we actually go through the division. When we think about cell metabolism and energy, we have to talk about where the cells get their energy from. So we have to break down fats, proteins, carbohydrates from the food we eat and convert them into the energy form of ATP. And ATP is adenosine triphosphate. When we talk about metabolism, it is the catabolism, or breakdown, of stored nutrients in the body tissues to produce energy. And we can do this through both aerobic and anaerobic ways. So aerobic is dependent on oxygen, while anaerobic is independent of oxygen. Aerobic is the one that our body uses the most because it's very efficient. We get a lot more energy out of aerobic respiration than we do for anaerobic. So that's catabolism. With anabolism, it's a constructive process. So catabolism is breaking things down, where anabolism is making things. So it's making more complex molecules from simpler ones. When we think about things passing through that cell membrane, we have to talk about different kinds of transport. We have passive and active. So our passive transport is labeled as passive simply because it does not require any energy. So you're not going to use any of your ATP in order to do this. So diffusion is moving across the membrane from a higher concentration to a lower concentration. And this can be anything moving across. Osmosis is moving from high to low, but osmosis only ever talks about the movement of water. And facilitated diffusion is diffusion, so it is moving across the membrane from high to low, but it has to be facilitated by something. So maybe there's a channel that has to open, maybe there's it has to be bound to something before it crosses. So in some way it has to be helped in order to cross that membrane, but in a way that does not require ATP. In active transport, we are going to be using ATP. So in primary active transport, we use ATP, we move it across the membrane. In secondary active transport, we're using another mechanism, either a symport or an antiport. So with a symport, think of symbiotic. Two things are helping each other. So with a symport, you may have two things moving across the membrane simultaneously. In antiport, they're going to move in opposite directions. So essentially you're exchanging. So you're exchanging sodium for potassium, but you have to use ATP to do it, because again, this is active transport. Lastly, we have endocytosis and exocytosis. So in these processes, it's not that something's crossing the membrane as we've just discussed and as we'll see in a moment. Instead, this is phagocytosis. So phage for eat, cyto for cell, and osis for process. So this is a process of a cell eating. And so essentially what happens is the cell engulfs, so it moves around and just swallows whatever it is it's trying to eat, and it moves inside the cell in that way. So it's not passing through the membrane. Instead, the cell is encompassing whatever it is it's trying to eat. The opposite is true for exocytosis. Again, it's not moving through the membrane, but now you have this little bubble inside the cell that's going to merge with the cell membrane and then release its contents on the outside. And we'll see examples of that as we move along. But just a reminder here, the prefix endo means inside, so we're taking something inside, where exo means outside. So endocytosis is the process of taking something inside a cell, where exocytosis is the process of moving something outside the cell. So this is what it looks like. So again, diffusion, everything moving through happily. Osmosis, water moving through. Here's facilitated diffusion, so something has to bind to a carrier protein in order to have that movement across. Here we see active transport, and this is an antiporter, so you can see that we're having sodium leave the cell while potassium's coming in. And here's pinocytosis. So pinocytosis, pino for small, and so what you're seeing here is that you're having substances come into this little tiny bubble and then move through the membrane in that way. Lastly, I need to talk a little bit about membrane potential. And this is really as a review for those who took phys, and a bit of a heads up for those who didn't. When we talk about membrane potential, essentially what we're talking about are gradients. And by gradients, I mean a difference in something on either side of the membrane. Whether it's a difference in chemical concentration on either side, or whether it's an actual difference in electrical charge. So the outside of the cell is more positive and the inside of the cell is more negative. So this membrane potential is really a huge important theme, especially when we talk about the nervous system, but also in the muscular system, and even in cardiovascular. So with the membrane potential, we have to talk about our ion channels. So our ion channels, again, are integral proteins, and we now know that those span the width of the membrane. We have protein subunits here that undergo conformational changes to open the ion channel and allow passage of things like sodium, potassium, calcium, or chloride. So essentially, those ion channels are squeezed shut or closed. Maybe they have a cap on them. So they're not allowing anything through. So when they are activated, which can be due to a number of different things, including a neurotransmitter, a chemical, or just a voltage change, when they undergo that change, now that channel is open, it's patent, and things can pass through. So an electrical potential exists across the membranes as a result of ion distribution. So again, if we have more positive charge outside and negative charge inside, then that gives us polarity, right? That gives us the idea of that membrane potential. There's a potential for change. So we need to describe the ability of separated electrical charges of opposite polarity. And this is what's going to allow us to do the work within a neuron and within the muscle tissue. So this leads us to the idea of the resting membrane potential. So the resting membrane potential is that amount of change on either side of the membrane. So this is a diffusion potential that describes the voltage generated by ions that diffuse across the membrane. Equilibrium potential is one in which there's no net movement of ions because the diffusion of because diffusion and electrical forces are exactly balanced. So essentially, things are equal on either side of the membrane, and therefore nothing moves. So again, you have to go back to the idea of diffusion, because diffusion, by definition, is the movement of something across a membrane from a high concentration to a low concentration. And so that diffusion wants to balance the concentration on either side, where our electrical forces are trying to balance that charge, right? Rather than having negative and positive, it wants neutral across the board. So diffusion potential is talking about that change in voltage generated by ions moving. Equilibrium potential is talking about what happens when everything is balanced. So that is a very brief introduction into membrane potential. But again, if you haven't had this in phys, it might be something you want to dive into a little bit more before moving forward. But now I have to, again, just very briefly talk about the tissue types. And you would have all have done this in anatomy anyway. But just the idea that cells with similar embryonic origin or function are organized together into larger functional units called tissues. These tissues associate together to form organs, and we have our 4 types. We have our epithelial, our muscle, our connective, and our neural, and those are the only 4 tissue types that we have. So with that, these 2 chapters are done, and the next video we'll get into the more meaty stuff, which is chapter 5 and chapter 8. So I'll see you there.